I have an agricultural background, and I've worked mostly throughout my life with sheep as an experimental model. So all of the data about which I will tell you today comes from this species. First, I wish to acknowledge the people who have contributed to the data that I will show you today. This is a list of the people specifically relating to today's data. James Cummins was a neurosurgeon friend of mine who helped me in my early days, and I'll show you some of that data. I have had 2 fellows in the laboratory recently who have played a very important role, some students, and a number of collaborators, some of whom, as you can see, come from Europe. And finally, my funding is these 2 sources. So, as Philippe mentioned, a long time ago, before some people in this room were born even, we generated these data which essentially show the pulsatile secretion of GnRH and how it drives the reproductive axis. These pulses of GnRH from the hypothalamus are the means by which there is communication to the pituitary and on into the outside world. The reason I really want to have this slide as my first data presentation is that this was essentially the very final proof of the neurohumoral theory of Geoffrey Harris, after whom this lecture is named. He had a number of stipulations for neurohumoral transmission. The most important was that there was a signal coming from the brain to the pituitary gland. And it was with the help of Jim Cummins that we generated these data. So this is the imprint of reproduction, but of course it's a lot more important than that, and there are a number of players upstairs in the brain who have significant impact. As we've found more and more in recent years, significant effect of energy balance, an impact of stress on the reproductive axis, and for animals like sheep, an effect of season, for which I will show you some data. The title of my talk has been is meant to mean that there is significant interaction with all of these factors into the reproductive system. So there's a considerable amount of crosstalk from within the body and from the environment. So, between 1982, when we first generated the data on the relationship between GnRH and LH secretion, a lot of work was done, of course, by reproductive neuroendocrinologists, but it didn't really change the textbook that much. Mostly, this was the model with which we were working, the GnRH neurons driving the pituitary gonadal axis, and here you see the female reproductive cycle. We knew that there was feedback effects of steroids on steroid-responsive neurons because, of course, these cells don't have the relevant receptors, and we knew that there were cells that connected to the GnRH neurons, but we didn't know what they were. And most of us were publishing data on the flavor of the month, whatever was the most popular neuron or peptide that had been discovered, but none of them seemed to really fit the model. What we did know, however, like all physiological systems, this is a closed loop, but we didn't have this particular component of the loop. So the most seminal findings that changed the whole field of reproductive neuroendocrinology came along in 2003 with these 2 papers, this one from Paris and this one from Boston, identifying the Kisspeptin system as being fundamentally important in reproduction in such a way that any mutation either in the receptor or the gene would lead to failure. What we showed and others have showed is that Kisspeptin, in fact, is a very potent stimulator of Gnrh secretion, and it became proposed very quickly that the feedback effect of steroids was via the Kisspeptin cells onto the Gnrh cells. So the first part of my talk then is going to be devoted substantially to the role of Kisspeptin in reproductive neuroendocrinology and what we have learned in very quick summary since 2003. These are Kisspeptin cells seen in the ovine brain. There are two populations, one in the arcuate nucleus at the base of the brain and a smaller population of cells in the lateral preoptic area closer to the cell bodies of Gnrh neurons. The thing that made these cells of immediate interest to myself and all reproductive neuroendocrinologists was that in the arcuate nucleus, at least, almost without exclusion, every Kisspeptin cell expresses estrogen receptor alpha. And we had never previously found a population of cells with such a high degree of exclusivity. Interestingly, in the preoptic area, the population of Kisspeptin cells there often, in fact, at least in half of cases, does not express the receptor. So, immediately, a very simple model was constructed whereby it was thought that the sex steroid feedback effects, which are fundamental to driving the female reproductive cycle, were mediated by the arcuate nucleus and the preoptic area Kisspeptin neurons that then transmitted the signal to the Gnrh neurons. But of course, this is far too easy. And hopefully in the course of the next 20-odd minutes, I'll explain why. The first problem is that the Kisspeptin cells of the arcuate nucleus also express dynorphin and neurokinin B. And here you see these Kisspeptin cells nicely producing NKB, and here dynorphin as well with the merge on the extreme right. Appropriately then, these cells became known as the Kisspeptin neurokinin B dynorphin or KINDY cells. And this was actually work that was first done by Bob Goodman on sabbatical in my lab in 2006. Now, what is the significance of this co-expression of these peptides within the same cell? Well, it turns out that the cells, these KINDY cells, are talking to themselves, in fact. NKB exerts a paracrine effect on Kisspeptin neurons, as demonstrated here, a feedback effect either at the level of the cell body or the terminal, and a very recent paper by Young et al. This year showed quite nicely that if you take mutated human populations in which the NKB gene or the receptor is mutated, you can totally restore reproductive function with Kisspeptin administration, short-circuiting this paracrine effect. So these cells essentially need the paracrine effect of neurokinin B. Let's move on to a brief discussion then of the feedback effects of sex steroids on the Kisspeptin cells. Here you see in situ hybridization in the arcuate nucleus, the third ventricle, for KINDY cells, which are profoundly upregulated by the simple act of gonadectomy, ovariectomy, and downregulated again by estrogen replacement. So the long-term negative feedback effect here, and as demonstrated in all other species, appears to be relayed through these KINDY cells of the arcuate nucleus. That is the long-term negative feedback effect which pertains for both estrogen and progesterone. But what about the positive feedback effect that drives the Gnrh surge and the ovulatory LH surge. This is thought to be caused by elevated levels of estrogen feeding back to the brain to cause a Gnrh surge and then onwards to the pituitary for LH. But is Kisspeptin involved? Well, certainly we know that the arcuate nucleus, at least in this species, is the site of positive feedback. And when one goes to that nucleus, either during the luteal phase of the estrous cycle or in the late follicular phase, just at the time when the LH positive feedback is being initiated, you see, especially in the mid and caudal regions of the arcuate nucleus, upregulation of Kisspeptin gene expression. You also see upregulation of the preoptic population of Kisspeptin cells. So quite substantive evidence of a fundamental involvement of these Kindy cells in the initiation of positive feedback. Now, the model we are going to discuss on a couple of occasions in this talk is a very simple model of positive feedback, which is an overrectomized ewe given a single injection of estrogen, which elicits a time-delayed LH surge, very similar to that seen in the estrous cycle. This delayed response is presumed to be due to neural transmission, and going to the earlier talk about what is more wonderful, certainly we don't know what is involved in this neural transmission. But what we do know is that if we look in these cells in the arcuate nucleus for FOS staining in Kindy cells, and we look under three conditions, one being ovariectomy here, one being the negative feedback effect of estrogen that I showed you earlier, but notice here, within 1 hour of an injection of estrogen, the profound upregulation of the Kindy cells of the mid to caudal arcuate nucleus. Further evidence of a fundamental involvement of Kisspeptin in this very important ovulatory event. On top of which, in another study done by Gloria Hoffman and colleagues, it was seen that during the time of the LH surge, when GnRH neurons show a high level of FOS expression or activation, you see exactly the same thing in the preoptic Kisspeptin neurons. So these cells are activated at the time of the LH surge. So they facilitate the Gnrh LH surge. Remember, the FOS in the Kindy cells was at 1 hour, way back here, not shown in this data set, which is the initiation of the surge. So that remains somewhat mysterious. Final proof for any sort of endocrine mechanism is always well demonstrated when one has access to an antagonist, and one such antagonist for Kisspeptin was generated by Bob Miller and his colleagues. So going to this positive feedback model that I mentioned earlier, estrogen-induced LH surges with central administration of vehicle, but when Kisspeptin antagonist is administered, see a profound diminution of this surge. In this case, you see a somewhat delayed surge. Over here are the mean data set. Notice, in fact, that elimination of Kisspeptin only diminishes this response by 50%. So there are many other players. There are many other systems about which I could talk if I had time that also contribute to this event, not solely Kisspeptin. So the model then that we have now for this positive feedback effect is an arcuate nucleus event that is an initiation, a potentiation of the event in the preoptic area leading to Gnrh secretion and the LH surge. So now we have completely, I think, to some extent, explained how this feedback effect of estrogen occurs, although we don't know the mechanisms and the cellular details. But there is another way, and kidney cells may whisper to the GnRH terminals within the median eminence. So as we know, the GnRH neurons project into the neurosecretory zone of the median eminence, seen here in a higher power, and these dark areas are the hypophyseal portal blood vessels that absolutely fascinated Geoffrey Harris. What fascinates us is that when you double stain with Kisspeptin, you see a very dense neuronal population in intimate association with the GnRH terminals. So if one takes the median eminence from a sheep, put it in a dish, and administer Kisspeptin, you see a very nice stimulation of Gnrh secretion. So this is another way that Kisspeptin may be interacting in the reproductive axis. Now, one of the useful things about working with sheep is that some of the times they breed and some of the times they don't breed. They're short-day breeders, and it turns out that kissing depends on season, such that the expression of Kisspeptin neurons is much higher in the breeding season than is seen in the non-breeding season. So it's always nice confirmation if one has some stimulatory system to see that it conforms to this seasonal pattern of breeding. So who else is talking at this Chatterbox Cafe? What are the other, and I put this in for the Americans, especially those who listen to the Prairie Home Companion, because there's a lot of chatter that goes on there. I want to talk a little about other neuropeptides that affect GnRH and LH secretion. And here you see an appetite-regulating peptide and a potent orexogen, neuropeptide Y, causing profound reduction in both LH, well here in fact LH, but also in GnRH secretion. On the other hand, if one infuses a melanocortin agonist into the cerebral ventricles of a sheep during the luteal phase of the cycle, you see the opposite effect, in fact, a stimulatory event. Now, melanocortins, of course, inhibit appetite. And one of the general rules that has come from this work and many other data sets is that everything that affects appetite also affects reproduction. General rule 2 is that orexogens inhibit reproduction and anorexogens stimulate reproduction. And that's been shown for many other neuropeptides about which I don't have time to discuss today, but I simply here show you some data indicating how Kisspeptin input is clearly demonstrated on a melanocortin cell. And of course, this cell is within the arcuate nucleus, so these cells are in close regional contact, on top of which, in an NPY cell, you see intimate associations of Kisspeptin input. Using these data, we've been able, and other data sets for neural tracing, etc., we've been able to construct neural wiring circuits in such a way to indicate the association between Kisspeptin cells and neuropeptide Y cells, POMC cells, etc. So that there is some means of transmission through this system in terms of metabolic regulation as well as reproductive regulation. The nice thing about this model, of course, is that factors coming from the periphery, such as estrogen or progesterone, may, first of all, signal to the Kisspeptin neurons that then can transmit effects onto the metabolic axis, and vice versa, how leptin and insulin may do the reverse. I could spend a long time talking about Kisspeptin, and of course we spent a whole week talking about it in Japan just recently at a congress devoted to the single peptide, which I thought was rather phenomenal. But what I do want to spend the rest of my talk discussing is what I've called an interjector. So this moiety is interjecting into what we understood to be the classical pathways regulating GnRH secretion, and many textbooks will tell you that you only need GnRH to regulate reproduction. But what I'd ask you to do is remember the growth hormone axis, of course, and how readily they accept somatostatin. So what I'm going to talk about now is gonadotropin inhibitory hormone, which negatively regulates reproduction. So this was identified in the Japanese quail by Professor Tetsui in the year 2000, and for a long time people produced some data in mammals that was not really well accepted over this period of 6 years. But then more recently, data began to emerge that convincingly demonstrated that this peptide inhibits reproductive function in mammals. It's an interesting gene. It encodes for 2 peptides within the same gene. I'm going to be talking exclusively about RFRP3. Notice it's only 8 amino acids in length. Quite a short, but as I will show you, quite an impressive little baby. It is produced exclusively in the dorsomedial nucleus of the hypothalamus, in a fairly well circumscribed group of cells. These cells project, as demonstrated here, to GnRH cells, and once again, usefully, we employ our breeding season model, and here you see a reduction in the number of the total number of GnRH cells during the breeding season, and you also see a reduction in the number of these contacts in the breeding season. So this then is the removal of an inhibitory factor, which is permissive of activation. Most surprising to us, but I'm sure it would have been very pleasing to Geoffrey Harris, we found terminals containing GnIH in the neurosecretory zone of the median eminence. And once again, here you see the portal blood vessels. And I'm sure you're well convinced that this is a fairly dense plexus of neurosecretory terminals. So, does this act on the pituitary gland to affect LH secretion? Well, using in vitro cell cultures, we show here stimulation of LH secretion by GnRH at 10 to the minus 9 molar, but notice with increasing concentrations of GnIH overlaying this GnRH, you see graded reduction in secretion of LH. Using an in vivo model of an isolated pituitary gland, we also show that in response to a GnRH stimulus, GnIH infusion reduces the response in terms of LH secretion. In terms of mechanism, it does this by acting within the gonadotropes. So here you see FURA2 signaling. This is the mobilization of calcium by GnRH. After 30 minutes treatment with RFRP3 or GnIH3, you totally eliminate that response to GnRH. So this is a potent pituitary effect. So of course then, the next logical question would be whether or not it is secreted into the portal blood, and indeed it is. So these levels of GnRH in the portal blood, here in the periphery as a control. So this is an indication of active secretion, and pleasingly, we see increased secretion during the non-breeding season. So this is inhibition of reproduction by this inhibitor being secreted into the portal blood, interestingly, to act on the pituitary gonadotropes. How does it relate to the events surrounding ovulation? Well, turning once again to cycling animals here during the luteal phase of the estrous cycle and the late follicular phase of the cycle, you see in situ hybridization through the rostral, mid, and caudal regions of the dorsomedial nucleus, and you see consistent suppression of gene expression for GnRH in the period leading up to ovulation. So once again, this is the removal of an inhibitor which is permissive of stimulation. On the other hand, of course, if you go to the late follicular phase and infuse GnRH, intravenous, mind, acting on the pituitary gland to totally eliminate LH secretion. Now, as I showed you earlier, Bob Miller went to a lot of trouble and probably spent an awful lot of money to generate a Kisspeptin antagonist as a means of suppressing reproduction, but he didn't need to do that. He just needed this naturally occurring GnRH molecule, 8 amino acids. So when this is infused across the period of the expected estrogen-induced LH surge, you see profound suppression of the surge event. This is, again, an intravenous treatment, mind, so it implies a pituitary action. Let me turn, finally, to one of the things that has intrigued reproductive physiologists and reproductive neuroendocrinologists for many, many years, and that is the issue of how stress impacts on GnRH and gonadotropin secretion in a negative way. There are many data in the literature, most recently a series of papers from Fred Karsh's lab showing how cortisol has a negative impact, but there is no indication of pathway or mechanism as to how this might occur. So we asked the question as to whether cortisol might act on GnIH neurons, and certainly they possess the relevant receptors, and whether that might be the means by which GnRH neurons are suppressed by a stress event. Now, we're not allowed to go around stressing animals for days and weeks and months, so what we did was we used a model of chronic pseudostress. Daily synapthin depot injections which elevated cortisol levels for around 6 hours every day. And what you see is a significant elevation of the level of gene expression for GnIH in the dorsomedial nucleus. So for the first time, I think we have a pathway or a conduit or a means by which we can investigate how stress negatively impacts on the reproductive process. So I stress then that this is an effect of chronic cortisol, and we've yet to do the studies to see whether there are effects in the acute mode and whether that impacts on the secretion of GnIH into the portal system. But don't worry, we'll do them post-haste. So just then, finally, I see my time is up. This simple model then that was originally proposed by Harris and others of neurosecretion of hypophysiotropic factors, in this case GnRH, has proven to be true, but it is subject to a lot of crosstalk, a lot of chatter, and a lot of interjection from higher brain centers. So the Kindi cells of the arcuate nucleus then regulate GnRH neurons. We know that there is at least one interneuron here about which I haven't been able to talk today. I've shown you data indicating action at the terminals, which I think is an intriguing line of investigation for the future. Also, what I never mentioned today is that there is another layer, of course. Glutamate and GABA cells input these Kindi cells and, of course, may be the true pacemakers of the system. The preoptic Kisspeptin neurons I mentioned briefly that facilitate the positive feedback effect of estrogen, and there are a whole range of other inputs to the cell bodies from the noradrenergic pathways from the brainstem and other systems that I haven't mentioned either. What I have mentioned is GnIH, which I've shown acts at the level of the cell body, but most intriguingly has been found to be the only neuropeptide, in fact, that has been discovered to come down the hypophyseal portal system for the last 20 years. So I think Geoffrey would be quite pleased about that. So this is the new textbook, I think, of reproductive neuroendocrinology, and it's certainly changed very significantly in my lifetime. And I hope that this brief vignette has given you some update on that. That's all I have for you today. Thank you very much.